Welcome to the Deep Dive. Today, we're cutting through all the noise to get to the core of, well, a really complex field in biology, plant reproductive physiology.
It's a huge one.
It is. We've pulled together findings from a major research volume that connects everything from genetics and hormones to morphology and even, large-scale ecology. We're going to try and give you the full picture of how plants actually pull this off.
And our mission today is pretty ambitious, too. We're trying to distill these incredibly specific scientific findings into something you can really use, something digestible.
Right.
So for anyone studying botany or maybe working in agriculture, or frankly, if you're just fascinated by how life works, this deep dive it's your shortcut.
And the stakes are just so high. I mean, you think about it, so much of our civilization is built on plants getting this right. Every seed we eat, every crop we propagate through grafting or cloning.
Oh, absolutely. If the fundamentals go wrong, you're looking at massive agricultural failure. And you can see that in the background of the volume's editors. You have people like Dr. Philip Stewart, who's a deep expert in genetics. He actually patented a strawberry plant.
Really?
Yeah, it's called Drisstraw 7. It came directly out of his work on flowering genetics. And then you have Professor Sabine Globig, who kind of bridges plant physiology with a background in human anatomy.
Which is an interesting combination.
It is, but it shows you how universal some of these biological strategies really are, even across different kingdoms of life.
Okay, so let's unpack this. Let's start at the very, very beginning, the molecular level, the instructions for making reproductive cells, the gametes. We can start on the male side, maybe with the model plant, Arabidopsis thaliana.
And the key player there is a transcription factor called DUO pollen 1, or DUO1.
DUO1. Wow.
What's so fascinating about DUO1 is that it's a single protein. Specifically, it's an R2-R3 mybed protein, and it has to juggle two totally different but equally essential roles for the male germline to actually work.
A single protein doing double duty. How does it manage that?
Well, first, DUO1 is absolutely critical for what's called gamete differentiation. It's like a master switch that turns on all the other genes needed for fertilization.
Okay.
Genes for cell communication, for fusion, things like ADMGH3, ATGEEX2. It's basically telling the cell what its final job is, its destiny.
So it tells the sperm cell what it needs to become.
Precisely. But then, second, it also regulates the cell division itself. It controls this critical cell cycle factor called ATCypB11. And without that, the cell just rusts. It can't complete the division needed to make the two functional sperm cells.
Which you need for double fertilization.
You absolutely need it. So if you have a mutant, the DUO1 mutant, does it just fail to divide, or is its identity wrong too?
That's a good question.
It's actually both. Both go wrong, which is what proves it has this dual function. The DUO1 mutant is completely sterile.
Wow.
And researchers tried to fix it. They could sort of partially rescue the cell division part by, artificially forcing the expression of that ATCypB11 gene. But even then, the cells, they still couldn't do their job.
So they divided, but they didn't know what to do after that.
Exactly. They failed at their purpose. It shows that the differentiation role of DUO1 is totally separate and just as critical. It's a really beautiful example of evolutionary efficiency. One switch for both fate and function.
Okay, so let's shift to the female side. The embryo sac is, it's famously difficult to get to, right? Tucked away deep inside the flower. How on earth do you study the genes being expressed in there?
Yeah, it's a nightmare. They use a really clever technique. It involved genetic subtraction and micro or array profiling in Arabidopsis.
Okay, break that down.
So they compared normal wild-type plants to these two sporophytic mutants, Co and Splee, which can't form a functional embryo sac. Then they basically digitally subtracted all the genes expressed in the sterile mutants from the data of the wild type.
So what's left over should, in theory, be the genes only active in the embryo sac.
That's the idea. It's a powerful filtering technique. They ended up with about 1,260 potential embryo sac genes.
And what did they find? Anything interesting?
Well, as you'd probably guess with this kind of novel research, the biggest category, about 35 to 37%, was unknown function.
Of course. Always the biggest category in biology, isn't it?
It really is. But they did find some functional examples. For instance, they found a gene called PUP3. It's a purine permese, and it was specifically expressed in the synergid cells.
And synergid cells are? They're the helper cells, right? They guide the pollen tube.
That's right. They're right next to the egg cell, and they're critical for guiding the pollen tube to its final destination. So, finding a purine transporter specifically located right there, it suggests some kind of unique metabolic or maybe a signaling role in that final critical guidance phase.
So we've covered the molecular building blocks. Let's move up a level to the architecture, how the whole reproductive organ gets built and, how that's timed. This usually comes down to hormones, doesn't it?
A very complex dance of hormones. A great example is stamen filament development in Arabidopsis. How does the plant make sure the stamen is the right length at exactly the right time?
It seems like something that would be very easy to get wrong.
It is, and it works like a relay race between hormones. It starts with gibberellin, or GA, a classic plant growth hormone. Its first job is to repress a set of growth-inhibiting proteins.
Proteins.
Nadella proteins, exactly. Specifically RGA and RGL2. You can think of the DELAs as the molecular brakes on growth. GA comes in and basically takes the foot off the brake.
Okay, so GA removes the inhibitor, but that's just the first step, I'm guessing.
Exactly. GA doesn't do the actual work of elongation itself. The next step is fascinating. GA's action then ramps up the production of a second hormone, jasmonate, or JA.
How does it do that?
It activates the genes that make JA, genes like DAD1 and LOX1. So you have a GA signal that leads directly to the synthesis of JA.
And why is the jasmonate so important here?
Because JA is the hormone that actually gives the command for the structural change. The JA that gets produced then triggers this wave of expression in specific transcription factors, the MYB proteins.
MYB21, MYB24, and 57.
Those are the ones. These MYD proteins are the indispensable engineers that physically drive the stamen filament to elongate.
Yeah.
So you have GA influencing JA, which then activates the MYB structural genes. If any link in that chain breaks, the whole thing fails.
Wow. That complexity.
Yeah.
It shows how just a tiny shift in environmental timing could have these huge cascading effects on the flower's architecture.
Absolutely.
So speaking of architecture, what about the whole flower structure? Floral symmetry. How is that controlled, especially in really specialized flowers?
Let's use the example of Opisthandra dincucianensis. It has a highly zygomorphic flower.
Meaning it has bilateral symmetry, like a face, not radial like a daisy.
Exactly, like an orchid or a snapdragon.
Yeah.
This particular species only has 2 fertile stamens, and the research shows the symmetry is controlled by a set of genes called CYC-like genes.
And what do these CYC-like genes do? Do they, promote the 2 stamens that make it?
It's actually the opposite. They act as repressors. They actively suppress stamen development in certain positions, which leads to the abortion of the dorsal and ventral stamens.
So they're molecular stop signs.
Perfect analogy. They're stop signs. And they do it by negatively regulating another set of genes, the D3 type cyclins. Since cyclins normally promote cell proliferation, shutting them down ensures those extra stamens just never develop properly.
Which brings us to the application of all this, timing, seasons, perennial plants. I was fascinated by the strawberry research in the source material, especially the stuff about ever-bearing genotypes.
Oh, it's a fantastic piece of applied horticulture. I mean, for a perennial strawberry, the challenge is linking flowering time to these really complex cycles of juvenility, dormancy, chilling.
All of it.
All of it. And what they found in these ever-bearing genotypes is that flowering is actually delayed by short days and low temperatures.
That sounds completely backwards.
It's the complete opposite of what you see in traditional short-day strawberry types, which use those same cues to trigger flowering.
So the ever-bearing ones are holding back when conditions get tough.
Exactly. It suggests there's this really robust genetic safeguard in place. It's likely tied to carbohydrate reserves, cold hardiness, something that tells the plant, hey, wait for better times. And understanding that is critical for breeding more resilient perennial crops.
Okay, so once the architecture is perfect and the timing's right, the plant has to get its gamete package, the pollen, where it needs to go. Let's talk about the pollen itself, its structure, how it's packaged.
The structure of the pollen grain, its ornamentation, is a beautiful example of coevolution. In the Araceae family, the aroids, they found a really strong correlation. The spiky or acinulate ornamentation only showed up in species that were pollinated by flies.
So the spikiness literally evolved for the fly.
That's the inference, yeah. And what's really interesting is that the ancestral pollination strategy for aroids was probably beetles.
And they don't have the spiky pollen.
No correlation at all. So it suggests a relatively recent, very clear evolutionary adaptation to maximize how well the pollen sticks to flies.
And not all pollen is shed as individual grains, right? What's the advantage of something like pollen cohesion, like in the custard apple?
Right. Annona chirimula sheds its pollen in little groups of 4 called tetrads. And the advantage is, well, it's adaptive. It makes the whole package more robust for transfer, and you're potentially delivering a bigger payload of pollen at once.
And how do they stick together? Is it like a glue?
It's more of a developmental tweak. It's all about timing. There's a delay in the normal process where the mother cell wall and the tapetal chamber dissolve to free the individual microspores.
So it holds them together for longer.
Exactly. That delay gives the microspores time to rotate and physically stick to each other with these things called pectin cohesion bridges. Then the hard outer layer, the sporopollenin, gets deposited and locks them together for good.
It's just incredible. A tiny adjustment in cell wall timing creates this huge ecological advantage. Which brings us to the final step, the communication. Pollen tube guidance. How does the tube find its target?
Okay, so this relies on this really elegant 2-part chemical signaling system. First, the pollen tubes have to gain what's called targeting competence. They have to be ready to listen for the signals.
And that happens when they land on the stigma.
When they interact with the stigma and style, yeah. Then they start listening. And what they're listening for is a signal from the unfertilized ovule. It releases this diffusible, heat-labile attractant.
A chemical beacon.
A chemical beacon. It's a relatively large molecule that creates a gradient, and the pollen tube just follows that gradient all the way home.
Okay, that gets the first one in. But you don't want 2 or 3 tubes getting in. That would be polyspermy, right? Catastrophic.
Catastrophic. So that's where the defense mechanism kicks in, the polyspermy block. As soon as one pollen tube successfully targets the ovule immediately switches its signal.
From come here to go away.
Precisely. It releases a much smaller, short-range repellent signal. And this thing causes any other approaching pollen tubes to just stall. They stop growing or they turn sharply away. And the key is speed. This happens in as little as 10 minutes.
10 minutes. That's incredible. Okay, let's zoom out completely. We've gone from the DUO1 protein all the way to this rapid-fire chemical warfare. How do all these external pressures ecologies shape these reproductive strategies?
Yeah.
Let's start with sexual selection.
Yeah, it's fascinating to see sexual selection theory, which we usually think of with animals, applied to plants. But it holds up. In plants pollinated by animals, males are competing for the pollinator's attention.
And the female's success is limited by resources.
Exactly. And the study on the moth-pollinated saline latifolia proved this beautifully. The male flowers were actually smaller than the female flowers, but they pumped out significantly more floral scent.
So they invested in advertising.
Heavy advertising, especially the specific scent compounds that are known to attract moths. They're trying to maximize their chances of getting their pollen out there. Meanwhile, the female, who has to invest all that energy into making seeds, puts her resources into structure, not perfume.
Makes perfect sense.
Yeah.
Okay, what about life history trade-offs? Like, why would a plant ever choose to self-pollinate, to self, when outcrossing gives you more genetic diversity?
This comes down to the time limitation hypothesis. The idea is that if you're in a really temporary or high-stress habitat, speed is everything.
Get it done before you die.
Get it done before you die. And when they compared selfing and outcrossing annuals, they found the selfers had significantly shorter bud development time, and their flowers didn't last as long.
They're cutting corners to save time?
They are actively shaving time off the whole cycle just to make sure they set seed before mortality hits. It's choosing immediate success over long-term genetic variation.
Okay, and what about physical threats, like being eaten? What's the worst kind of damage for a prairie perennial?
The simulated herbivory study was really clear on this. Meristem removal was way more damaging than just defoliation, than just losing leaves.
The meristems being the growing tips.
Right, the growing tips. They have the stem cells for all new growth, and critically for new flowers. So if you lose your leaves, you lose your solar panels, which is bad. But if you lose your meristems,
You use the factory itself.
you lose the factory. It severely reduced the number of flowers in plants like phlox and rosa. And while heavy leaf removal did reduce the total seed weight, removing the meristem just fundamentally wrecked the plant's ability to even try to reproduce in the first place.
Finally, let's look at the biggest scale, habitat fragmentation. What happens when we chop these ecosystems into little pieces?
The findings from the Brazilian Atlantic forest are, well, They're stark. Fragmentation just leads to a collapse in the complexity of pollination strategies.
How so?
All the specialized systems, the ones that rely on specific birds or flies or even non-flying mammals, they were either lost completely or just drastically reduced.
And you can measure that loss.
Oh yeah. The remaining fragments had a significantly lower functional diversity index. Pollination systems were down by over 30%, floral types down by almost 24%, floral sizes down by 21%.
Right.
It means the smaller fragments they're getting dominated by generalist pollinated species. You're losing this huge, measurable, intricate web of specialized relationships that really defines a healthy ecosystem.
We have covered so much ground today. I mean,
Yes.
from a single protein, DUO1, managing both cell fate and division, all the way to the devastation caused by fragmentation.
Yeah.
Through hormonal relays, rapid-fire chemical signals,
Yeah.
it's just mind-bogglingly complex.
And what really stands out is the integration, how it all connects across every single one of those scales. A plant's ability to manage its timing, to specify its cells so precisely, and to respond to everything, internal signals, external pressures.
It's the whole ballgame.
That's the whole ballgame. It's the foundation of agricultural stability, and it's the absolute focus of conservation efforts around the world.
Well, thank you for joining us for this really intensive deep dive into plant reproduction.
My pleasure. And for those of you listening, especially self-taught learners or students thinking about plant breeding, here's a little challenge for you to think about.
Okay.
Consider the MET1 gene in Arabidopsis. We know it affects seed size, but it has opposite effects depending on whether you manipulate it in the father's gamete tissue or the mother's somatic tissue. So the question is, if you're a breeder and you want to reliably increase seed size for a bigger yield, which path do you take? Do you reduce MET1 activity in the father's gametophyte, or do you reduce its dosage in the mother's integuments? And more importantly, why? It'll force you to connect that molecular regulation with the really critical differences between maternal and paternal contributions to seed development.